So we talked about hormones before in here, and so you probably know what they are, but just to make sure, hormones refer to chemical signals that are released by a plant, or by any cell, I guess. And what they do is they bind to receptors that are receptor proteins that are embedded in the cell membrane, and then those cause something else to happen inside the cell that we refer to as a signal transduction pathway, and that's going to change the behavior of the cell. So chemical signals that bind to receptor proteins embedded in the cell membrane. The binding of hormones causes a signal transduction pathway to begin. It changes the cell's behavior. So, in humans, our behavior of the overall animal or overall body is mostly mediated by nerves, right? We certainly have hormones, but the quick stuff, the being able to move around and walk and stuff like that is all handled by your nerves. You do have hormones as well that cause long-term changes, right? That are not as fast acting, and then you also have this like hybrid of hormone/nerve, where the hormones begin to interact with the nerves in something that we call neurotransmission. They can act as a neurotransmitter and as a hormone at the same time. But in plants, they don't have nerves at all, and so all of the responses are slow. Okay, every plant response, with the exception of something that's called nastic movement, is slow. And nastic movement is not based on hormones. Nastic movement is based on turgor pressure. It's based on the rapid change in cellular pressure, or the amount of water pressure inside the cell. So that means that all plant responses are going to be relatively slow compared to animal responses because they're doing them with hormones. They're not doing them with nerves. Okay. The most important thing for you guys to understand is that these are classes of hormones, and the classes of hormones can do a lot of different things, and some of the mechanisms are well understood, and some of them are very not well understood. Okay, so we're going to talk about some of the ones that we understand better first. Auxin is a good place to start. Okay. Auxin is sometimes called indole acetic acid. That's its chemical name. And sometimes they abbreviate its chemical name as IAA. So sometimes they'll try to be tricky on the AP exam, and they'll tell you, they'll give you a reading, and the reading will be about indole acetic acid and its function on plants. And you'll be like, I don't know what indole acetic acid is. Well, it's just auxin. And so you'll be like, oh, well, I do remember what auxin does. I know how it functions. So you can sort of have a leg up if you recognize that. Okay. We know that in high concentrations, auxin is going to inhibit cell growth, right, and cell division. And the place that we talked about this was in that thing that's called apical dominance. Which means that if there's an apical meristem around, that apical meristem will produce auxin, and that will stop all of the axillary buds that are close to it from being able to start to grow. We said this is useful because it stops the plant from becoming heavy at the ends and drooping down. Right, so this is our auxin concentration. High concentration here will inhibit the growth in these axillary buds, but these axillary buds are actually stimulated by the medium auxin concentration. Okay, so that means that in medium concentrations, Auxin stimulates growth. And then it talked about in the terms of phototropism as well. And phototropism is interesting because those cells have already grown. They've already divided. They're not in a meristem. And so there's no way for them to make more cells. So they have to get by with having the same amount of cells, and they have to be able to move the plant. So they're not making new cells. They're just changing the cells in order to make the plant move. And the way that that works is by elongating the plant. Okay, and the way that elongation happens is by increasing auxin concentration, which softens the cell wall, right? So the cell wall is made out of cellulose, and if you can soften the linkages between those cellulose molecules, then you can allow for the plant to stretch out more as its central vacuole fills up with water, right? So say that auxin can promote cell elongation by softening the cellulose cell wall and allowing the central vacuole to stretch the cell. Yeah, so we'll talk about that more tomorrow, but phototropism is essentially the ability for plants to grow towards light, and the way that that works is that let's say that there's a light source like over here, right? This plant will then have auxin build up on this side, right, which causes the cells on this side to elongate, which pushes the plant towards the light source because this side is longer than this side, and it's caused by auxin buildup. So auxin builds up over here, which weakens the cell walls, which causes the cells to expand, which then pushes the plant that way. Yeah. Vacuole. Yeah. All right. So let's talk about the next class, the Gibberellins. Oh, before we move on, I always say that if you're answering a question on plant hormones and they ask you specifically to name like what type of hormone is responsible for this this mechanism in plants, and you don't know, it's not like one of the ones you automatically know, you should just answer auxin, because auxin does like 90% of the things in plants, varying concentrations of auxin. So it's a pretty safe bet, okay? Let's talk about the Gibberellins. These are sometimes called gibberellic acids. Or sometimes they're called just GA. They promote stem elongation. And they also promote fruit growth. So they promote fruit growth in two ways. Number one, they can cause a unfertilized ovary, unfertilized fruit ovary, to begin fruiting, right? Or flower ovary to begin fruiting. So normally what happens is a plant makes a flower, right? Then down in the base of the flower there are the eggs, the ovaries that are producing ovum, right? And then the pollen comes in and gets trapped by the flower, and then it grows a pollen tube down, and then it fertilizes the egg that's in the receptacle of that flower, right? And then the flower dies, and now you've got a fertilized egg, a zygote in here. Those develop into seeds, and then a fruit develops around it. Okay, that's how normal fruiting happens. But the fruiting response is controlled by gibberellins. Gibberellic acid is naturally produced whenever fertilization occurs and there's a zygote in the ovary, right? So what you can do is, if you don't want to have a seed in your fruit, you can just spray the plant with a lot, or spray the flowers with a lot of gibberellic acids, and they get the message that all of their ovaries have been fertilized, even though none of them have, right? And they'll just start fruiting. Okay, so seedless grapes, for instance, if you've ever had like seeded grapes, you know that there's a big hard seed in the middle of most grapes, of those type of grapes, but they don't really even sell those anymore because they're annoying to eat, right? And you can very easily just make seedless grapes by spraying the grape vine with gibberellic acid, and then they'll start fruiting. They do this with a lot of different plants. They also do it with watermelons, right? Although one of the things that they also do with watermelons is they speed up the process of the growth of the fruit, because that allows for the seeds inside to not be developed. That's why they're the little white seeds instead of the black seeds, because those seeds haven't had time to mature. But anyways, this is a natural response. A natural plant response would be to produce the gibberellins whenever there's fertilization that occurs, but we just give them the gibberellins without fertilization, which causes the fruiting response, which leads to seedless plants and stuff, or seedless fruit and stuff. All right, let's talk about the cytokinins. Cytokinins have one obvious mechanism, one obvious function, and that is that they promote cytokinesis, right? Because they're called cytokinins. Which is cell division. So you would expect there to be a lot of cytokinins in the meristems, and there are, right? Because they're promoting cell division. The other place that's weird, that we don't necessarily understand the mechanism behind it, is we know that cytokinins spike in concentration right before a tree or a plant dies from old age, right? Some trees have a lifespan. You know, some of them are in the like 70 to 80 years. Some of them are significantly shorter than that, and some of them are longer than we know. Like we know that the sequoias out in Sequoia National Park, some of them are thousands of years old, right? So what makes something live 300 years or makes something live 80 years? Well, if you could figure that out, you'd be very, very rich, because the mystery of aging is one of the things in biology that we just don't get. We don't understand why certain things die at certain times. But one of the things that we do understand, or we do see at least, is that while a tree is in this process of dying from old age, the concentrations of cytokinins go through the roof, right? So we don't necessarily know if it's causing it or if it's a byproduct of it or something like that, but we know that they're present in high concentrations when a plant is dying due to old age. All right, let's talk about abscisic acid, or ABA. Abscisic acid was named because we thought that its main role was to promote what's called leaf abscission, which means leaves being dropped during the wintertime. Okay, so leaf drop. But further experiments have told us that this is probably not the case. We know that it's present when leaves are dropping, but it's likely that it is present due to the fact that the leaves are dropping, not that the leaves are dropping due to its presence, right? And the way that we know this is because you can have, if you have a mutated tree that does not produce abscisic acid, it can still drop its leaves. and it can do it without abscisic acid, which kind of flies in the face of it. And the other thing that we can figure out is that if you have two trees that are, let's say, they're saplings or something like that, and they're in pots, right? And one of them begins to drop its leaves. If you take those two pots and you put them close together, the one that's dropping its leaves makes the other one start dropping its leaves, right? So they can like sort of communicate and say like, hey, I'm dropping my leaves, you should do that too, okay? And that's weird because if it was abscisic acid that's doing that, abscisic acid is an acid, so it's water soluble, and that would mean that they're sharing water soluble stuff, right, which we would think would happen through the soil, but if they're in two separate pots, there's no way that they could do that, right? So it's very likely that it's controlled by another thing that's called ethylene gas that we'll talk about in a second. One of the very well understood mechanisms of abscisic acid, though, we'll say not well understood here. It's probably part of the process, but it's definitely not the thing that initiates the process. But one of the things that we definitely know abscisic acid is important for is promoting what's called seed dormancy. Okay, so seeds generally found inside fruits. Okay, so you know, an animal eats the fruit and takes it to someplace else, maybe spits out the seed, maybe poops out the seed, whatever, right? It doesn't matter, but the seed gets distributed somewhere else. Okay, generally speaking, it's not good for that seed to immediately start to germinate, right? It wants to wait until the conditions are good for it to germinate. It wants to, you know, make sure that it's going to have enough sunlight, enough water, that the temperatures are going to be right so that it can have the greatest chance of being able to survive, okay? And the thing that helps it do that is abscisic acid, okay? So abscisic acid is an acid, right? So that means that it's water soluble. And because it's water soluble, that means that the more water you expose it to, right, the more that water is going, or the more the abscisic acid is going to dissolve into the water, and the concentration of abscisic acid inside of the seed is going to decrease, okay? So if you go to the store and you buy a bag of lima beans, a bag of dried lima beans, okay? Those lima beans, you can put them in your pantry. You can wait 10 years. They will never, ever germinate. They're seeds, right? But they're not going to germinate. But you take those same lima beans, you throw them into a cup of water for like, you know, a couple of hours, 4 or 5 hours, right? Then you pour out the water and you refill it, right? Do the same thing, pour out the water and refill it. And what happens is that abscisic acid that's inside of the seed is leaching out into that cup of water, and then you pour it out and you put fresh water in, and it leaches out more abscisic acid. Pour it out, put fresh water in, more abscisic acid leaks out. And now you can just take that lima bean and just like throw it on a paper towel, and it'll start to germinate. Okay? The thing that was preventing it from germinating was the abscisic acid, and as the seed took in water, it washed away the abscisic acid from the inside of it, and that then allowed it to germinate when the concentration of abscisic acid became small enough. So we can say that as seeds take in water, that's called imbibation. The ABA washes away, allowing the seed to germinate. That's why when you're planting seeds, they always say you should soak them first. You should always take them and soak them into a bucket of water. And that's the other thing that when I was, you know, a kid and I would go and do a little gardening experiment or something like that, right? I never understood because it'll say like on the packet of seeds, let's say I'm gonna grow strawberries or something like that. On the packet of seeds, it'll say like in a 5-gallon bucket, fill a 5-gallon bucket with water and place the seeds into a 5-gallon bucket. I'm like, well, that seems stupid. I have like this many seeds. Why would I put it in a 5-gallon bucket? Why don't I just get like a mug or something like that and put it in there? Well, it's because the 5-gallon bucket has a greater concentration of water, or more water, and that means that the concentration of abscisic acid can go down far enough, right? It can wash away, it can dilute into that bucket of water. Whereas if you just have a little mug, right, that mug is going to become concentrated with abscisic acid, and that's not necessarily going to wash enough of it out of the seeds, right? So in order to do it in a mug, you have to do it over the course of a couple times. You have to put water in, dump it out, put water in, dump it out, yada yada. But if you just do it in a big bucket, then you don't have to worry about that because there's enough water for it to wash away. All right, let's talk about ethylene gas, last one. Ethylene gas is a gas, obviously. That's one of the unique things about it. It promotes leaf drop. We talked about that. So if you take a plant that's dropping its leaves and you put it next to another plant that's not dropping its leaves, the other plant will begin dropping its leaves due to the presence of ethylene gas, right? The other thing that it does is it ripens fruit. It promotes fruit ripening. Okay, fruit ripening is different than fruit growth, right? Fruit growth, we said, was stimulated by things like gibberellic acid, whereas fruit ripening refers to taking the complex carbohydrates that are in there, the starches and stuff, and breaking them down into monosaccharides and disaccharides, right? Because starches, we know if you ate like a potato or something like that, let's say you just go to the store and you buy a potato and you just chomp into it like an apple, right? It's not going to taste super good. It's going to be really starchy and it tastes weird, right? But if you chomped into an apple, it tastes good. Well, what's the difference? The starches that are in the apple have had time to break down back into smaller sugars like disaccharides and monosaccharides, and that gives them their sweet taste. And as the ripening continues, they get sweeter and sweeter and sweeter and sweeter and sweeter, right? Because there's more monosaccharides and disaccharides and less of those insoluble fibers in there. Also means, interestingly enough, that the calorie content of fruits changes based on their ripeness, right? So if you eat a fruit that's like still really crispy and not very ripe, it's got a lot of fiber in it that your body can't digest, right? But you give it time for the enzymes that are inside of the fruit to break down that fruit into monosaccharides and disaccharides, and then your body can process them, right? And so the calorie content goes up, and so the sweeter it is, the more calories it has, because that means that your body is going to be able to utilize them. There's some interesting things here that you can do with fruit ripening. You've probably heard this before. So if you have like a bushel of bananas that are nice and green, and you're like, man, I wish I could have a banana today. If you take that banana, a banana off the stack, and you put it in with a overly ripe, like disgusting black banana, right? And you put it in a bag for like an hour or two, it'll ripen the other banana, because the more ripe a banana gets, the more ethylene gas it produces. And so if you take an overly ripe banana and put it into a bag with a not ripe banana, it'll make the not ripe banana start to be the right ripeness. Okay, and you can do this with all fruits. Bananas are just one of the ones that we use as an example because they produce a significant quantity of it. But if you took a banana and you put it in with like a peach or something like that, the peach would get really, 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 really ripe in a really short period of time. Like over the course of 2 or 3 hours, it might get to the point where you couldn't even eat it anymore. It would be so soft and squishy, right? But this is a way that you can control the ripening of fruits. And so when we ship like a giant shipment of bananas from some South American country to the United States to be eaten, right? We have to put them in container ships, right? The big metal containers. And the way that they stop all of the bananas from ripening in the container ships is by putting a giant fan on one side of it and a giant fan on the other side that forces air through. And by forcing the air through, they're pulling in fresh air and they're pushing out the ethylene gas, right, to stop the bananas from ripening. But if they didn't do that and they were just all sitting in there, as the second one of the bushels started to ripen, it would cause ripening in all the other bushels, which would mean that you'd just get a bunch of rotten bananas by the time they got here. But if you can stop the ethylene gas from building up, then you can prevent that from happening. I also saw a really cool infomercial. It was like one of those 2 a.m. infomercials for like this fruit saver bags, these like plastic bags that you could put fruit in, and they were like, put it in the fruit saver bag and you can keep a banana at the correct ripeness for 2 weeks. And I was like, that doesn't seem possible. So I went online and I looked at their patents, And one of the things that they have is it's a plastic that is coated in a catalyst that breaks down ethylene gas and absorbs it, right? And so as the banana starts to release ethylene gas, right, it gets absorbed by the package, which stops it from ripening any further. And it really does stop it from ripening because then there's no ethylene gas present in that confined space. It's also the reason why if you've ever tried to buy a fruit basket, most fruit baskets are open on the sides because you don't want the ethylene gas to start pooling in this, closed off fruit basket, because then all of your fruit will start to get overly ripe at the same time. But if you have it in an open basket, it just kind of leaks out onto your countertop and disperses, right? Anybody have any question with that? That's it.